As the global population ages, the burden of age-related diseases continues to rise, putting immense pressure on healthcare systems and society. Scientists are increasingly interested in ways to slow down the aging process, not just to extend lifespan but to improve the quality of those extra years. Interventions that target aging could help prevent or delay the onset of chronic diseases, offering significant benefits to individuals and communities. However, measuring the biological aging process in real people has remained a challenge, limiting our ability to test which interventions truly work. This is where new approaches like using wearable devices and machine learning come into play, opening up exciting possibilities for accessible, personalized health insights. Unlike chronological age, which simply counts the years since birth, biological age reflects how well your body is functioning relative to your peers. It's influenced by genetics, lifestyle, nutrition, and environmental factors. Two people of the same chronological age can have very different biological ages depending on their health and habits. Scientists have developed various ways to estimate biological age, such as measuring telomere length, gene expression, or metabolomics. However, most of these methods require invasive tests or lab work, making them less accessible to the general public. The search for a simple, non-invasive way to measure biological age has led researchers to explore new technologies. Wearable devices like fitness trackers and smartwatches have become incredibly popular, collecting detailed movement data from millions of people every day. These devices can track steps, activity intensity, and even sleep patterns, providing a rich source of information about daily life. Researchers realized that this data could be used to estimate biological age, since mobility and movement are closely linked to overall health and vitality. By analyzing patterns in movement data, scientists hope to develop models that can predict biological age in a way that's accessible to anyone with a wearable device. To bring this idea to life, researchers developed the MoveAge model, which uses machine learning to predict biological age from wearable device movement data. They used large, publicly available datasets from the NHANES study, which included accelerometer readings from thousands of adults. The model analyzes features like maximum activity intensity and movement variance throughout the day, summarizing a week's worth of data for each person. By training a random forest algorithm on this data, the team was able to predict an individual's age based on their movement patterns, opening the door to new ways of tracking aging in real time. The NHANES datasets from 2003-2004 and 2005-2006 provided a unique opportunity for this research. Thousands of participants wore accelerometers on their upper thighs for a full week, generating minute-by-minute movement data. The researchers filtered the data to ensure high quality, including only adults with complete, calibrated records. This resulted in two robust datasets, each covering a wide age range from 18 to over 85 years old. These datasets not only included movement data, but also demographic, nutritional, and prescription drug information, allowing for a comprehensive analysis of factors influencing biological aging. To make sense of the vast amount of movement data, the team focused on two key summary statistics: the maximum intensity of activity for each hour and the variance in movement during each hour. These features capture both the vitality and diversity of a person's daily movements. For example, younger individuals tend to have higher maximum intensities and more varied movement patterns, while older adults often show lower values in both. By reducing the data to these summary measures, the researchers could efficiently train their machine learning model without losing important information about physical activity and health. The MoveAge model was built using a random forest algorithm, a powerful machine learning technique that combines multiple decision trees to improve prediction accuracy. The researchers split the 2003-2004 dataset into training and testing sets, using 70% for training and 30% for validation. By feeding the summarized movement data into the model, they taught it to recognize patterns associated with different ages. The model's performance was then tested on the reserved validation set, showing a strong correlation between predicted and actual ages. This approach allowed the team to create a reliable tool for estimating biological age from wearable data. After training, the MoveAge model was validated using the 2005-2006 NHANES dataset. The initial predictions showed a good correlation with chronological age, but the model tended to overestimate ages for younger people and underestimate for older individuals. To address this, the researchers normalized the predictions by comparing each person's predicted age to the median predicted age of their age group, then adjusting accordingly. This normalization improved the model's accuracy, achieving a correlation of 0.93 and reducing the root mean square error to just over 8 years. The result was a robust, reliable predictor of biological age from movement data. One of the key outputs of the MoveAge model is delta age, the difference between a person's predicted biological age and their actual chronological age. A positive delta age indicates accelerated aging, while a negative value suggests decelerated or healthy aging. By analyzing delta age across the population, researchers could identify individuals who were aging faster or slower than expected. This metric provides a practical way to assess the effectiveness of interventions and lifestyle choices, and to identify people at higher risk for age-related health problems. The MoveAge model revealed a strong link between accelerated biological aging and higher mortality. In the NHANES dataset, individuals with positive delta ages, meaning their biological age was higher than their chronological age, were significantly more likely to have died during the follow-up period. This association was especially pronounced in people over 40 and remained significant into their 70s. Conversely, those with negative delta ages had lower mortality rates. This finding confirms that movement-based biological age is a meaningful indicator of overall health and longevity. Digging deeper, the researchers examined whether certain causes of death were more closely associated with accelerated aging. They found that chronic lower respiratory diseases, such as COPD, showed the strongest trend toward higher delta ages. This suggests that reduced movement capacity, possibly due to respiratory problems, is reflected in the biological age predicted by the MoveAge model. While other causes of death did not show significant associations, this insight highlights the potential of movement-based age prediction for identifying specific health risks. One of the most exciting applications of the MoveAge model is its ability to identify lifestyle factors associated with healthy aging. By linking dietary intake data from NHANES to delta age, the researchers searched for nutrients that correlated with decelerated aging. They analyzed dozens of dietary components across different age groups, looking for patterns that might reveal which foods help slow the aging process. This approach offers a powerful, real-world way to screen for potential geroprotective nutrients in large populations. The analysis revealed that certain nutrients became increasingly important for decelerated aging as people got older. In particular, higher intakes of fiber, magnesium, and vitamin E were strongly associated with lower biological ages, especially in people in their 50s and 60s. These findings align with previous research showing that fiber reduces mortality risk, magnesium deficiency is linked to age-related diseases, and vitamin E may support longevity. The results suggest that focusing on these nutrients could help promote healthy aging, especially later in life. Beyond nutrition, the MoveAge model also enabled a retrospective screening for pharmaceutical drugs associated with decelerated aging. By comparing delta ages of drug users over age 70 to non-users, the researchers identified compounds that might slow the aging process. This approach generated a ranked list of drugs, highlighting those with the strongest associations to lower biological age. While most drugs showed no significant effect, one stood out as a potential geroprotector: doxazosin. Doxazosin, an FDA-approved alpha blocker commonly used to treat high blood pressure, emerged as the only drug significantly associated with decelerated aging in the analysis. Users of doxazosin had lower biological ages compared to their peers, suggesting a possible protective effect. However, association does not prove causation, so the researchers turned to laboratory experiments to test whether doxazosin could actually extend lifespan and health span in a controlled setting. To test the causal effects of doxazosin, the team used the nematode worm C. elegans, a well-established model for aging research. Worms were treated with doxazosin at different concentrations, and their movement and lifespan were measured. The results were striking. Worms given doxazosin showed significantly greater mobility in later life and lived longer than untreated controls. These findings provide experimental evidence that doxazosin can extend both health span and lifespan, at least in this model organism. Doxazosin works by blocking alpha-adrenergic receptors, which are involved in blood vessel contraction. In C. elegans, similar pathways are linked to stress resistance and protein homeostasis, both important for longevity. Suppressing these pathways can activate protective responses in cells, such as the unfolded protein response, which has been associated with extended lifespan in worms. While the exact mechanisms in humans remain to be clarified, these findings suggest intriguing biological pathways that could be targeted for healthy aging. While the MoveAge model offers exciting possibilities, it's important to recognize its limitations. The model may capture patterns of daily life such as work schedules rather than purely biological vitality, It might not fully reflect all aspects of health like mental decline or cancer risk. Additionally, associations found in population data do not always indicate causation. More research is needed to confirm whether interventions like increased fiber intake or doxazosin use truly slow aging in humans. Nonetheless, the model provides a valuable tool for large-scale, real-world screening. One promising implication of this research is the potential for personalized nutrition advice based on biological age. If certain nutrients are shown to slow aging, individuals with signs of accelerated aging could benefit from targeted dietary interventions. This approach could help slow health decline and improve quality of life as people age. As wearable devices become more widespread, integrating biological age prediction with personalized health recommendations could become a reality for many. The MoveAge model demonstrates the power of combining wearable technology, big data, and machine learning to advance aging research. As more data becomes available, models can be refined to include additional health markers and lifestyle factors. Future studies may explore how interventions like exercise medications or new diets impact biological age over time. Ultimately, these tools could help individuals and healthcare providers make informed choices to promote healthy, active aging. In summary, predicting biological age from wearable device movement data offers a practical, accessible way to monitor healthy aging. The MoveAge model links movement patterns to mortality risk and identifies nutritional and pharmacological factors associated with decelerated aging. While more research is needed to confirm these findings in humans, the approach opens up new possibilities for personalized health interventions and large-scale screening for geroprotective compounds. As technology continues to evolve, the dream of extending not just lifespan but health span moves closer to reality.